Okay, so I think we'll go ahead and get started. We have close to 100 people attending this webinar, so welcome everybody. My name is Omar Harb, and I will be presenting some parts of this webinar. Hopefully everybody can see the slide I'm showing. Before I get started, just some logistics. In your screen, in the GoToWebinar screen, you should have an option to raise your hand if you have a question. Also you can type your question. There's a question panel which allows you to type the question, and we will be monitoring the questions, and we'll answer them either by replying to the question itself or verbally if it warrants more general discussion. So feel free to use those two options, and we will continue monitoring those to make sure we get all the questions answered. This webinar will probably last about an hour, and our goal is to cover some basic aspects of VEuPathDB resources, in particular just some basics on how to get started, and then we will focus on some key features and functionalities of the database. This is not everything, obviously, but the webinar series, which I will show you towards the end of this webinar, will highlight some of the future webinars which will focus on some of these aspects in more detail. So there will be many more opportunities on a weekly basis to learn more about the tools and functionalities in VEuPathDB resources. Today, the presenters will be Evelina Bishenko, who's based at the University of Liverpool, and she will be covering the phenotype aspects of this webinar. I am Omar Harb, which I'm based at the University of Pennsylvania, and I will cover the basics, how to get started, some of the Galaxy tools, and wrap up towards the end. Sam Rund, who's joining us from the University of Notre Dame, will be covering MapVEu, and he will tell us more about that. And Suzanne Warrenfels, who's joining us from the University of Georgia, will be covering the RNAseq searches and going through the details of how to run these types of searches. So to start off with, let me take you here. So VEuPathDB, in case you haven't noticed, we are in the process of merging between the VEuPathDB, formerly VEuPathDB, and VectorBase. This includes a number of different components for vectors of human pathogens, but also for the parasites themselves and various important fungi. This process is ongoing, so there will be changes over the next few months, hopefully, where you will see a new interface that merges all of our resources. But for the time being, we're still, at least from a web interface, we're separate entities. So if you go to veupathDB.org, you will see an interface that looks like this. And if you go to VectorBase, you will see an interface that looks like this. All of these resources have different entry points, but as we merge, we will be developing a unified web interface that, so you will access the data and access the same functionalities on all of the websites. The cool thing about this merger is that we are taking the best of both worlds and putting together into one resource. And so hopefully the next few months will have some exciting things to show you. So to start off with any of these resources, usually there's an entry point, either with specific searches, or you may have a specific interest in a particular organism. So if you go to VectorBase, you have various tabs here that allow you to quickly navigate to the organism of interest. You can access various tools in the menu up here, or access the data itself if you want to download it or visualize it in more detail. There are obviously a lot of helpful information here in the central portion and different ways of accessing the data. But importantly, there's a search option at the top here, which allows you to type a word like kinase, for example, and clicking on go will allow you to get the results, but those results are also categorized on the left. So this is a very nice way for you to quickly navigate between looking at the results that you're finding across the entire vector-based database, but also zeroing in on to results that you're interested in. So for example, if you were only interested in the kinase results from Ixodes scapularis, for example, you can click on Ixodes scapularis now and your results will be filtered in the central portion right here. So this feature will actually be something that will be made available in the merged resource, and so that'll be very, very cool. If you were using the VEuPathDB resources, again, there are many different ways of starting your searches, including running a text search at the top here or adding a gene ID, but also a number of searches right here in the center, which are are specific to data types. And they're divided up into various categories. For example, all of the searches right here on the left and the center left are searches that return lists of genes. And the types of searches that Suzanne and Eve will be showing you a bit later on are specifically searches that are found in this category here and will return lists of genes. All of our resources are built on the same infrastructure, so they will all look the same. You can find the same searches there. And if I ran a search, for example, for kinase, you will get a result that looks like this. And this is called a search strategy. And so this is one of the first things you will experience when you run a search in our databases, is you will get a search strategy. And again, this is a feature that has been very popular that we will be providing for you in the integrated resource. The cool thing about this search strategy is that once you run your search, you get the results right here. For example, in this case, I ran the word kinase across all pathogens and organisms in VEuPathDB, and there are over 133,000 results in this case. And you can go down and look at the results if you want. But the real advantage of this strategy system is that it allows you to add steps and grow this experiment. This is an in silico experiment, basically, which allows you to grow the experiment and combine results from different searches. So for example, I can add a step. So this add step button is a very useful tool. And when I add a step, I get a pop-up which provides you with a list of all the available searches. And so for example, my first search looked for kinase. I can add another step and ask, well, I'm wondering how many of these kinases are potentially secreted because I have a secretory signal peptide. And if I click on protein targeting and localization, you'll notice that there's a predicted signal peptide search here. And once I click on this, you will be offered some options. So I can select which organisms I'm interested in. I can, for example, select all. If you're on one of the component databases, let's say PlasmoDB, you will only see Plasmodium organisms. But for now, I'm on the VEuPathDB portal, which gives you access to all the organisms. So I can say I want to find all organisms that have a signal peptide. And if I expand this, there's some advanced parameters. So you're welcome to play around with these advanced parameters if need be. And then I have to decide how I'm going to combine the results of the signal peptide search, which I'm running right now, and my previous results, which were for the word kinase. And I'm offered some options here. In my case, I'm interested in things that are a signal peptide and a kinase at the same time, right? So I'm looking for the intersect operation. And hopefully this little graphic here gives you an idea of what you're going to be returned. So I'm going to be returned the little green portion of this Venn diagram. And then I click on run step. And so what this does, it runs a second search. And the second search will look for all genes that have a predicted secretory signal peptide. And remember, this is searching hundreds of organisms. So the results may take a few seconds to load, but this wasn't too bad. So it found over 540,000 genes with a signal peptide, and the intersection is about 13,278 genes. So these are kinases with a predicted secretory signal peptide. The cool thing about this is that you can continue expanding the search as long as you want. And so asking, combining results from any of the available questions in our resources. Okay, so with this, I think this will be the short intro, hopefully getting you started and comfortable with using the resource. As I mentioned, we will have more targeted webinars in the future, which will focus on these different aspects, and I'll talk to you about them later on. One other note, we are recording this webinar, so this webinar will be available for you and others afterwards. So don't fear if something went by too quickly, you will have access to it afterwards. So I'm going to go ahead and switch over now to Suzanne Warrenfels, who's going to be telling us about RNA sequence queries. And of course, as I mentioned before, feel free to ask questions, enter comments in the question section. Actually, before I switch over, Suzanne, 1 second, sorry, I just wanted to mention one more thing. Is that one important aspect of our resources is that you are able to log in and create an account. While you don't have to log in to use the resource, logging in allows you to store your searches. And so it's almost like a lab book. So you can store them and come back to them later on. So I would highly encourage you to do that. In addition, we have a contact us link here, which allows you to send us an email and ask us questions. We love hearing from you. So if you have questions and concerns or things you are wondering about, send us, contact us, email, and we will respond within 48 hours usually. All right, with that, I'm going to switch over to Suzanne. All right, Suzanne, we can see your screen.
Okay, and you see just prioritized data?
Yes.
All right, sounds good. So I'd like to take a minute or two to share our ideas about integrating RNA-seq data, and then I'll move into a discussion of the searches. We want to produce the most useful resource for our research community, so we take quite a bit of care in prioritizing data. We watch the literature closely, discuss with the researchers and experts in the field, and take suggestions from the community to find data that's pertinent in the field. And our experiments that we integrate span a wide range of designs. So we have time courses, for example, that define the entire life cycle. We compare virulent and avirulent strains in some of them. And then we also have studies like knockout studies that show the effects of specific genes on transcription. Most of our data is associated with publications. It's a very rare event that there's not a publication associated with a data set. Sometimes we do get data pre-publication, but then eventually it's published. And so we pull data from a repository. Researchers deposit their data in a public read archive, and then we pull the data from there. And these raw data are also available to you. Right, and then we are tasked with analyzing the data. We use the same workflows across all data sets so that you can compare results of all the experiments against each other. Our workflows eventually produce a table of expression values. And here's a little cartoon of just the very, very basics of what RNA-seq analysis is. So we start with the raw reads and do some quality assessment just to get rid of reads that didn't complete. And then we align that to a genome. Align those reads to a genome, and from this data we can say something about the transcript structure and abundance. And eventually we come up with a table of each gene's expression value in all samples for that experiment. Right, and then we build ways for you to search that without actually having to do this yourself. So I'm going to demonstrate some of these searches in PlasmoDB. Right, and the RNA-seq searches are categorized under search for genes based on transcriptomics and RNA-seq evidence. You can also reach this page from this drop-down menu up here. So, and right away you can see that there's not just one search. This page tabulates 25 or 30 available data sets in PlasmoDB, and you can use this table to choose the data set and the type of search that you want to do. So the first column is the organism that the data is aligned to. The second column is the data set name, and which does not always match the publication because many of the RNA-seq data sets are part of larger studies that include like microscopy and invasion assays and the publication title is just not appropriate to describe the RNA-seq data. So if you hover over these question marks, you get a summary of the actual data set and a quick link to the publication so you can investigate and find what data set you want to study. In the third column, then you have all of the searches. These are buttons that lead to the searches that analyze the specific data sets. So the data that I want to use today is from 2018 and is a comparison of ring oocyst and sporozoite transcriptomes. So I can look through this list to find it, or I can use this handy dandy filter up here. If I type ring, then I find these 10 or so data sets. And if I continue to look, I come up with just one, right? And you can see there's 4 types of searches available for this. And let's just define those quickly, right? DE is differential expression, and this experimental design allowed for statistical analysis of the data, so it had the proper number of replicates and so on, and we could apply statistics. The fold change is a great way of exploring fold change differences of genes between samples, especially when a statistical analysis is not available. So it's just really an algebraic calculation. The percentile, for the percentile searches, we applied a percentile rank to the expression values, and then we built a search that allows you to probe the expression based on distribution of the gene's expressions within a sample. And then last but not least, we have sense antisense, which looks for genes with simultaneous changes in sense and antisense transcripts, which may occur when antisense expression represses actually expression of the gene. Okay, so let's start off and show a differential expression search. I'm going to open that in a new window. And the search page opens with, a title at the top and parameters that you can use to define the search on the left there. We have a get answer button, which will initiate the search. And then below that, there's a description with some help information and a link to our dataset record with more detailed information about this dataset if you'd care to go there and more understanding. So let's just set this up. For the experiment, I'm going to look at the sense data, and I am going to use the sporozoite as a reference and compare that to the oocyst, looking for upregulated genes with a fold difference of 2 and an adjusted p-value of 0.005. How about that? Let's change that to 0.005. And I click Get Answer, and I'm taken to the result. So in the result, I can see I have 1,182 genes, and I think that might be too many to look at. So I can go back and actually edit my parameters, right? So if I click Edit, Revise, I can say, let's look for genes that have a 20-fold difference with, a 10-fold higher p-value, and click Run Step. And I end up with 243 genes. This looks like a normal amount to look at. So let's look at the structure of the results here. Up at the top, we have a graphic of your growing strategy. In the middle is a distribution table of your hits across all of the organisms that we searched. And we only actually searched one organism, 3D7, because that's what this data was aligned to. And then at the bottom, we have the gene results table, and that's just a list of all your hits. The first column is a link to the gene page, and then we have all kinds of associated data with that. So you can see that this second gene here had a fold change value of 872 and a p-value of -53 range. You can use this Add Step button to add any data that you would like to this table, including FPKM values from your from the actual experiment. So I'm going to search for that, and I find the experiment here, and I'm going to add normalized sample values for each of our samples, and then they're added to the table. So not only am I looking at fold change, but also the raw values too. Another thing to note is that you can download these results. You can add them to a basket, which would you can shuffle them off to other searches. But then there's also this new analyze results tab, where you can run on the fly basically enrichment analysis, and we offer a gene ontology enrichment, a metabolic pathway enrichment, and a word enrichment. Okay, so let's go back and and run the fold change search for this data set. I opened that in a new tab. And while it looks a little bit more complicated, it's really not. We've added some help information over here, which kind of squeezes things together. So for this experiment, I'm going to look for protein coding regions, protein coding genes that are upregulated with a fold change of 20 between sporozoite and oocyst. Basically the same search that I had set up in the differential expression. I click get answer, and I find that I have 710 genes, whereas before I had like 240. And that's because we have lost the parameter of a p-value here. So, but the advantage of this search is that you can group samples together in a way that you couldn't with the statistical analysis. So if I click revise, I can say I want to look for genes that are upregulated 20-fold between sporozoite and the average of expression between oocysts and rings. And you can see that this help image changes here. I'm going to average these two comparison samples and compare it to the reference sample. And when I do that, I come up with a fewer number of genes. So again, you have everything. You know, you can do a go enrichment, or you can add columns to look at that data. All right, so we have actually two more types of searches that I'd like to demo. So here we are back at the page. I'm going to click on the percentile search, and we'll run a percentile search on this data set. So the parameters are experiment, and I'll choose sense. I'm going to look for genes in the sporozoite sample that have an expression value in between the 80th and 100th percentile, right? And I click get answer, and I am returned 1,121 genes. And you can see that each of these in the sporozoite samples has a very high expression. The other ones are variable, of course. We didn't put any restrictions on those in our search. So this is a list of genes that are highly expressed in this sporozoite sample. Again, there's a wonderful advantage to this search, and that is we can go to edit and revise. I can actually group these samples together and look for genes that are highly expressed or low, if you choose that, across all of these samples, right? So I'm going to choose all of these samples, say I want to look for genes between the 80th and 100th percentile in all of these samples. I click run step, and all of my genes have high expression in these samples. And you can look, you know, you can see their histones and housekeeping genes and stuff like that. So that kind of all makes sense to me. This is, you know, a powerful search to look for genes according to the distribution of their expression within a sample. Okay, and then we have one more search. Let's go back to here, the sense antisense. So this one is a little bit more complicated in concept, and so I will scroll down to the help information, and we'll set up the search according to this example here. And this graph shows us that we're going to be able to find genes with a 3-fold increase in antisense expression at the same time has a 2.5-fold decrease in sense expression. So you could imply that antisense expression is repressing the sense expression. So I'm going to scroll back up and set up this search accordingly. So I'm going to find genes whose antisense transcript increase threefold, while the same gene's transcript decrease 2.5. Let's just say in sporozoite compared to oocyst, right? And I'm going to click Get Answer. And I find 5 genes. So these are genes whose antisense transcription might repress their repress expression of that gene. Okay, how am I doing on time? I just want to mention that there are some other, so those are the major types of RNA-seq searches that we have. There are some other ones that are, you know, sort of organism specific, depending on, you know, the biology of that organism. And of course, the kinetoplastids have a splice leader, right? So they produce, you know, a long transcript, which is actually spliced into smaller transcripts. We have a search that's specific to that. We also have some circadian rhythm data, which we have a search specific to that. And then in fungi, we have a couple of similarity searches. So you can return genes with a similar expression profile as your input gene in these similarity searches. I think that's it.
Great, thank you, Suzanne. You can go ahead and transfer over to Sam. We've been getting some questions. I think we've been answering them offline. Hopefully, I know a couple of us are answering at the same time. So far, I haven't seen any contradictory answers, which is good. But keep asking questions, and we will answer them as we can. And Sam, will turn it over to you.
Thanks, Omar. So the feature that I'm going to show is called, will be called MapVEu when the new VEuPath merger of we're calling VEuPath is formally completed. But I'm going to show you in its current iteration where it's still called BioMap on the vector-based website. And while initially we'll just be porting this feature over to vector-based, in the future we'll start to incorporate this new map that I'll be showing across the VEuPath sites where data exists that make it appropriate. So you can access it here on our website, vectorbase.org, and then population biology, and then I will be showing the BioMap. So this is our map. So this is MapVEu. And what's really cool is it's very efficient at finding data. So you can see we have data from around the world, and this is not genomic data usually. This is all of this data is field collected data, and then some of which then has omic work performed on it. As you can see here in the right-hand drawer, we have numerous kinds of data, including abundance data, so how many mosquitoes or vectors were collected in a given area, pathogen data, what were those vectors infected with, blood meal host, what was the source of the blood, did they bite a human, did they bite a dog, insecticide resistance phenotypes, these are when they take mosquitoes that they collect in the field and expose them to insecticide and monitor them to determine how susceptible or resistant they are to the given insecticide. And then finally, I'll show insecticide resistance genotypes. So here we are starting to bring in genetic data. So it tests the vector for the presence of a mutation that confers some sort of resistance to insecticide. Now you'll notice that some of these views, the ones I mentioned, have these little icons here. This means that we've created a special view to help visualize that kind of data. So I'll go here to abundance view. And you can also change views with this drop down. And I'll click optimize colors just to make it look prettier. And what's really fascinating about our system is that you can start to filter to find exactly the kind of data you want. So let's say I only want to see data that's in genus Anopheles, and we'll see what kind of data we have available. And you can start to, as you click on data, you'll notice that the points get higher and higher resolution. This point here has a thumbtack, which means it's the highest resolution data point we have. So this is a trap level result. But for example, here, keep clicking, we keep seeing higher and higher spatial resolution. When I click on a data point, I have a number of options that I can use here in the left-hand drawer. So this is just a simple pie chart to show the different percentages of mosquitoes that were collected at this location. But I can also use this specialized view-specific data visualization because this data is regular surveillance. It has a temporal basis. And it also, just like we bin data by location to make it easier, we also bin by time. So here is the yearly resolution, but this data was actually collected at a higher resolution. So I can call it up and be presented with higher temporally resolved data too. As part of this process, just like we do for all the genomic data, we do a large amount of curation of the data to standardize it so that it looks, all the data that's similar will look the same, have the same standardized collection types and all metadata fields. So you can see that for each and every record that we have, we've collected, for example, when it was collected, where it was collected, how it was collected, what attractants were used, and the results. So we spent a lot of time trying to collect very clean and standardized metadata. And in all, just for this abundance view, we have over 20 million individual records that have been collected. So it represents a lot of work, and we're really excited to bring this to our other communities within VEuPath. So I'll just quickly show some other specialized views. So this is our insecticide resistance view, and because the thing that is usually the most relevant information people want to know is how resistant are the mosquitoes here. So for this particular view, for example, we've added a color bar to show the degree of resistance to insecticide that the vectors have. And it's not just from the search bar the species that you can search for. So let's say I just want to see Aedes aegypti. In addition to the search bar, I can also use the right-hand drawer. Let's say I just want to see Aedes aegypti resistance to deltamethrin. And so I can see what kinds of data are available. And because this is a different view, that means that again, we'll see all the metadata, including what was the mortality rate, what was it exposed to, was it females or males, how was the test performed. So we have all that metadata. And because this is a different view, we're no longer in the abundance view. When we click on specialized view, we're going to see a very different kind of graph. So this is showing the results from the mosquitoes that were collected versus the background that we automatically compute based off of all the data in the database. And again, you can see the data from each and every point. And for any of our views, you can also download all the raw data. So you can do your own analysis, or a feature that we really like is, let's say you found a really neat graph, a really cool view that you want to share with your colleagues, and click on this button. That generates a link so that if you go, you can email that link to your colleague and they can call up and see what you were looking at. We have the same as I mentioned for pathogen data. So this is what the organism was infected with, what the vector was infected with. So I'll just show some examples here. So these are different plasmodia or viral infections of the mosquito. You can look at blood meal analysis. So what did the mosquito or the vector bite on? And finally, I will show a genotype view. which will probably be the similar to what we rolled out to VEuPath first, because it's the most relevant to the most number of communities. So what we have here are alleles that are known to be involved in insecticide resistance to mosquitoes. And we can search by a particular allele, like this, or I can just remove those search terms. Oops, sorry. Or you can, for example, look for a locus. So I will do, let's say this one. And then I can switch back to allele view. And I can see the breakdown in collections between individual mosquitoes that were collected showing specific alleles. And just like all the other views, we can zoom in and see, well, that was that one already had a thumbtack. I can zoom in and see higher spatially resolved data. I can look and see the metadata for all the collections that were made. I can download the data. I can share the data, the view, with a colleague. The final thing that I will show is I'll go back to Abundance View and just show that we do have some other kinds of searches that can be used to discover the data that you're looking for. You can filter by date range. You can filter by season. This might be relevant for malaria, for example, to just see what's happening in the rainy seasons, the high transmission seasons, or the dry seasons, depending on what you're doing. Oh, and actually I'll show one more thing. It's often important. We see a lot of these risk maps that are made, and you want to know how is that generated? How do they get that point? So for each and every point in our data, you can see exactly where that study came from. And if this doesn't give you enough information about exactly where that data point came from, you can go and see a whole page dedicated to explaining exactly how and where this data came from. With that, I will transfer to the next person.
Great, thank you, Sam. I think the next person is Eve. And we've been answering questions so far. There's one map question, so I'll leave that, Sam, to you to answer. You can go ahead. Actually, maybe Sam, I can ask you the question before Eve starts. I'll ask you the question verbally, and we'll see see if you can answer. If not, you can take it offline. But this map only shows mosquito-related data, not other vectors. Correct? Question mark. I'm looking for kissing bugs, for example, but can't find them in the list.
So we currently have data on mosquitoes and biting midges in the map, and I think a few tick results. We would happily put kissing bug data. That's definitely within our remit, and it's limited only by if people have submitted data sets to us or not. So we very much, if you know of a good data set, please send it our way, and we'd be happy to incorporate it.
Great, thank you. So let's go ahead and switch over to Eve. All yours.
All right, thanks, Amar. Hi, everyone. My name is Evelina Basinko. I'm a FindYouDB outreach, and today I will show you how to mine phenotypic data. I am showing essentially the landing page for FindYouDB, and we will initiate the search from the Search for Genes panel, which is shown right here. So the phenotypes can be accessed under the phenotype evidence submenu. And if you click on the link, it will essentially redirect you to a page that will show you a list of all datasets currently available in FungiDB for data mining. For example, we have several in-house manually curated datasets for fungal pathogens, Aspergillus, Cryptococcus, Fusarium, other organisms such as Neurospora crassa, and we have also integrated a PHI-based curated phenotype. For those of you who are not working with fungal pathogens and are not familiar, maybe not familiar with PHI-based, PHI is essentially a pathogen-host interaction database that curates data for host pathogens interactions. For example, they curate data for any gene that has a known association with, for example, disease in various pathogens. In this demo, we will use this dataset to set up a search that uses data from several species. So the use case that some of you may have, let's imagine for a second that we are researchers working with Candida auris. Candida auris is a fungal pathogen, and it's emerged in the past few years, and it's actually quite a formidable pathogen as it demonstrated a multiple drug-resistant phenotypes, and it is a worldwide health concern. So let's say you're a researcher and you would like to know or find any genes that may have an important role within the disease or may have important implication in clinical aspects of the disease. And as you notice, there are several Candida species available within this dataset. However, none of them are Candida auris. So what can you do? One way to set up your search is actually use FungiDB and the records it has for other organisms to mine this data and later on transfer your data into your organism of choice to make inference essentially for Candida auris. So let's go ahead and do this. What I would to deploy the search, I will click on the Curated Phenotypes button, and the dataset will be expanded on the bottom. That essentially brings up all the data that are characterized based on certain criteria. So let's go ahead and navigate to the pathogen species and select Candida organisms that are available here. You can technically go ahead and select one by one, but I will go ahead and search for Candida using the filter at the top to make sure that I highlight them all. And by clicking at the button here, it essentially will highlight for you all species that are listed here. Now that we have selected Candida species, let's go ahead and identify genes that are relevant for the disease phenotype that we're interested in looking at. I would like to identify all genes that have been annotated with either invasive or disseminated candidiasis. So let's go ahead and do this. There are several metadata options available. So I'll select any disseminated infections and also invasive candidiasis annotations. Okay, so it seems like we have two parameters, which will be pathogen species and disease. And within both of these, we identified certain criteria that now we would like to deploy the search. So let's go ahead and click answer. So as you notice, the page refreshed, and now you have arrived at the page that you have already seen in Suzanne's demonstration. It's essentially a step that says there are a number of genes available in the list below that fit your criteria. And again, if you have forgotten what the criteria were, you can quickly click on the edit button and it will bring up a pop-up screen with a reminder that you're looking for this type of infections and in these 4 pathogens. Okay, so the list at the bottom essentially has 144 genes, and this includes Candida albicans, Candida parapsilosis, and 2 other species. We could technically look at the individual genes located here. see if we can gather any interesting and important information. But we are researchers that work with Candida auris. We would like to know how many of these genes actually have orthologs in Candida auris. And there's a way to do this in all, essentially all of VEuPathDB sites. If you click on Add Step and then select Transform by Ethology step, you will deploy, or you'll actually bring up a pop-up screen to deploy another search. And this parameter essentially asks you what species you would like to transfer your data in, essentially. I will go ahead and use a filter to bring up Candida auris quickly, and click to select, and then click on Run step to deploy it. So here we are. You have now a strategy that consists of 2 steps. First step, identified all genes that have a specific disease phenotype. The second step is orthologous transformation step that now identify genes that are orthologous to the genes in step 1. Notice that there's also a change in the shape of the box that simply indicates that you are in the step 2, you're no longer working within the same genome that was selected in step 1. And if you scroll down a little bit further down the list, you will notice that you have a list of Candida auris genes with their genomic location, product description, and also their orthologs listed for Candida species. There are several additional columns that you can scroll down to the right to investigate further, and that tells you sort of a quick glimpse of how many paralogs or orthologs, if any, available for every specific gene within the list. And paralogs and orthologs are identified within the FungiDB database. There's also a link out to an ortholog group that will take you to the OrthoMCL site that will show not only orthologs in fungal species, but also in other parasites, including non-fungal pathogens. And I previously selected to look at the genes that have predicted transmembrane domains or signal peptides. And as always, you can modify the columns that you see by clicking on the Add Columns button. And you can actually turn off a few columns if you think this is not relevant to you, and potentially select a few more. So I'll go ahead and update to remove the 2 columns on the right. So now, going back to our search, just as a recap, we have looked at the genes associated with a disease phenotype of disseminated or invasive candidiasis in 4 Candida species, and then we have transformed this result into Candida auris, for which phenotypic data is not available yet. And for those of you who are interested in setting up additional queries, you can add step to bring up additional searches such as RNAseq data, or you can actually click on Edit Step and insert steps anywhere here, potentially exploring, again, RNAseq, microarray, proteomics data, or even SNP data, if available. Just a quick reminder, if you're interested in reviewing this strategy at a later time, you can find it under the public strategies lists. As Amar mentioned, you don't have to be signed in to the site to use the site, and public strategies will be available, even invisible, even if you do not have an account with us. But to navigate to the strategies, just click My Strategies, Public Strategies tab, and today's strategy that I have just covered with you is available right here. All you need to do is just click on the link, and it will be brought up as a strategy with additional description at the top to indicate what we have looked at, and further information will be available if you click on the View Description. And as always, if you have any questions about this strategy or any information in general, click on Contact Us link. And before I transfer this back to Amar, who will walk you through the Galaxy interface, I want to also mention that if you know any cool data sets that you think should be integrated in FungiDB or other VEuPathDB sites, let us know. click on the contact us link, send us relevant publications, and we will get back in touch with you. All right, Omar, back to you.
Great, thank you, Eve. So while you transfer the presentation to me, just a quick note. So thanks, Eve, for this portion. There've been a number of questions about different types of data. Why haven't you loaded this data, or when can we see this data, or do you have data sets for kissing bugs and so forth? So the bottom line is our goal is to provide you with access to data, and if there are data sets out there that you know of that you're not seeing in your favorite database for some reason, send us an email. There may be either we don't know about it, right, which is a possibility, or we're working on it and we are still in the process of developing the interface to provide the data. One of our philosophies at VEuPathDB is not to load the data to recapitulate exactly what the researchers found in the publication, right? So our goal is not to just remake the figures from the paper, because you can read the paper to see that, and you should read the paper and to see what the authors concluded and so forth. But our goal really is to provide you with tools to investigate and to interrogate the data a little bit more, take it a step further than the conclusions of the authors, right? So you should be able to recapitulate what they did in the paper, but importantly, you should be able to tune the knobs a little bit and find additional things that are interesting, that may be interesting to you and may have not been interesting to the original researchers. As we know, this genomic data is really deep. There's a lot of things to discover there, and not enough people to actually do the work on it. So I think there's more data to go around. The other thing that there was a very important question about whether I can, somebody was asking, can I run an RNA sequence question across all species or organisms in a particular database? And the answer is there isn't a universal query, because every RNA-seq experiment is really based on a specific experiment from a specific species. So we can't take that data and universally align it. That won't work. But this orthology tool that Eve just showed you, which allows you to transform a list of genes from one species to their orthologs or homologs in another species, is a really great way for you to jump from results from one species to another. So I can run a transcriptomic experiment, for example, looking at merozoites in Plasmodium falciparum, and then ask, what are the orthologs of these merozoid genes in falciparum in Plasmodium vivax? And then you can ask, okay, out of those vivax genes that are orthologs of Plasmodium genes, how many of them are expressed in this liver stage experiment from a vivax experiment, right? So this is a nice way for you to be able to combine results from diverse species, but using orthology as a link, as a way to transform from one species to the next. Another question also, just I think it's of general interest, and that is, we are obviously showing you examples in vector-based or examples in PlasmoDB or FungiDB, but all of these tools are available in all of our resources. So you can go to GiardiaDB, for example, and run an RNA sequence search, assuming the data exists, right, or we have it there, but the same type of search will be available there. So if there's a cyst versus a trophozoid RNA sequence dataset, which there is, or a proteomics dataset for that matter, you can go in and actually ask the same questions and say, give me any gene that is upregulated in trophozoites compared to cysts, or vice versa. Okay, so what I'm going to do for the remainder of this webinar is I'm going to quickly take you through our integrated Galaxy platform, where you can analyze your own primary data, and then wrap up. So all of our resources give you access to this section here called Analyze My Experiment, which is available to you at the top in the top banner section. When you click on Analyze My Experiment, this will take you to our Galaxy genomics website, or Galaxy platform interface. We didn't build Galaxy, right? Galaxy is built by another group. It's a very, very nice platform for people who are not who don't code, right, to be able to run many bioinformatics tools. And you will see, if you're not familiar with Galaxy, I would encourage you to check out some of the many tutorials that are out there about how to use Galaxy. But it allows you to really run all of these bioinformatic tools that are shown here on the left, and there are many more. These are not all of them. And the nice thing about this is you can run them using a web interface. So if you look in the methods section of a paper doing RNA sequence analysis, where they took the RNA sequence data and aligned it to a particular species using an alignment program called HiSat2, right? all you need to know really, HiSat2 is almost like a biological reagent that you use in the lab, right? It could be like your enzyme, that's your HiSat2, and you're going to put it under different conditions, whether it is at a 37 degrees for 2 hours and so forth. So these are your parameters. So in Galaxy, you can select HiSat2 and play around with the parameters and run the step on your data. And we have a number, when you first come to Galaxy, this is a workflow that I pre-ran, so you won't have this obviously, but you will have the central portion here, which gives you some example workflows. So another very nice advantage of Galaxy, it allows you to tether a number of tools together. So for example, in the lab, you may run an enzymatic reaction, then take your product out of the water bath, clean it up. So that's another step. Then you might take that cleaned up fraction and run it on a gel. So that's another step. You get your gel results, you take a picture of them. That's a third step, right? So in Galaxy, you can tether all of these steps together into one workflow. And so we have some predefined workflows for you here. And all of them take, for example, this is a workflow that takes paired and stranded reads and aligns them to a genome. If I click on this, it will give you a page where you can actually run this workflow. And the workflow includes a spot where you can select the data. So if I have loaded data in my workflow, you can select them. There are many more tutorials about how to do this, and we will be running a dedicated webinar just about Galaxy. So stay tuned if you're really interested in this. All of the organisms in VEuPathDB databases are preloaded, right? So this is a nice shortcut for you, so you don't have to preload the genomes and figure out what format they're in. So I can go in here and select the genome of interest, and then you can play around with all the parameters of this workflows. Whether you want to do a play around with the let's go down here. There's an HTC count, for example, parameter. There's a should be another one down here. So there's anyway, it doesn't matter. There are a number of different parameters here that you can, or tools that you can play around with the parameters for. Oh, here's HiSat. I was looking for this. So you can play around with the parameters of HiSat, and then you run this workflow. And once you run it, it's going to run all the steps sequentially, right? It'll take the results from one. Once it's done, it'll run the next step and the third step. And in the end, you will get a workflow that looks like what you have here on the left, which includes the results, which you can explore here, you can download. And one thing that's really cool that we have developed for you is a way for you to transfer the results back to your favorite VEuPathDB database. So we can use these export tools right here on the left. For example, export VEuPathDB, and you can export RNA sequence data to VEuPathDB. And we're developing a number of new tools here to export all sorts of kinds of data back to VEuPathDB. And once you do this, you can fill up this form basically, click on execute. And the results of this, in this case, this is a Plasmodium experiment. It'll be transferred back to PlasmoDB. And then I can go to my dataset section right here. And this will load all the datasets that you've been working on. So here's a list of datasets that I use for demonstration purposes on a regular basis. So this is my datasets. This is not public, this is private, right? So it's your own private workspace for your primary data. I can click on this differentiation experiment, for example. I can share the data with other people. As you can see here, I've shared it with other people. It tells you something about the experiment. And then it tells you that there's an available search for this experiment. If I click on this available search, this will show you a pre-configured search page, which is configured based on your data. And again, this is private. This is exactly what Suzanne was showing you earlier. So it's a way for me to say, well, I want to look for genes that are upregulated by, let's say, 10-fold in my female gametocytes compared to my male gametocytes. In this case, I took an experiment that I found on the sequence read archive, and I can change how I would like to compare them to each other, right? So I'm comparing these red ones to these blue ones, and I want them to be 10-fold, I should use a 10 here, 10-fold differentially regulated or upregulated, and I click on get answer. And so what this does, it will return a list of genes that met that criteria from my own experiment, right? Remember, this is your own primary data, and it will show you the list of genes. It will generate a custom graph based on your own data. So you can see, oh yeah, this gene looks like it's in the 3 replicates, it's upregulated compared to my other ones. You can do a Go enrichment on this data, or you can start adding steps and asking, well, in my experiment, these are the genes that were upregulated in female gametocytes. I wonder if these same genes were also upregulated in an experiment from another lab that was deposited in PlasmoDB. And so you can start combining these and asking some really nice integrated questions. Okay, so we're going to end here by, first of all, thank you everybody for joining this webinar. I hope you found it useful. As I mentioned, this webinar has been recorded, and you will get an email with access to the webinar link, which you can share with others. It'll also be available in our YouTube channel. We will also send you this link here, which is a link to our upcoming webinars. We are working on a series of webinars that we'll be presenting to you, and please feel free to join these as you see fit. This will take you to a page that looks like this. So we have right now 6 webinars that are, or today's webinar is obviously done, but there are 5 additional webinars that are scheduled, and we'll continue adding more here. So next week we'll have a webinar that will introduce you to the genome browser in VEuPathDB resources. We didn't talk about the genome browser today, but we have a genome browser that allows you to look at the data in the database in a browser interface. We will, in the beginning of May, we'll have a webinar that will go do an in-depth tutorial on how to run a search strategy, combining searches, orthology, and so forth. On May 14th, we'll have a webinar that's dedicated to talking about Galaxy workflows. So we'll talk about how you can construct a Galaxy and workflow, how you can run it, and what the results will look like. And finally, so far on May 21st, we have a webinar that will take you through manual gene annotation using the Apollo interface, which is a very exciting interface that we're bringing from Vectorbase into the family of VEuPathDB databases that will allow you to go in and change gene models based on evidence and your knowledge. And so that's an exciting opportunity. And as I mentioned, we will add many more webinars as time goes on. All right, so we will end here. I'm going to just double check and see if there are any last minute questions. Just many thanks. Thank you everybody for attending. If there are any questions that we missed, we will try and answer them offline, but I think we got all of them. So thank you very much again for attending, and see you next time, hopefully. And stay safe and healthy. Bye-bye.
Bye.
Bye-bye, guys.